आपण डायल केलेला नंबर.
That one is not open, sir. Not open.
Not open.
Hello?
हाँ संध्या.
हाँ, okay, sir. Okay, sir.
Okay ना?
हाँ, yes, sir. Yes, sir.
Actually,
हाँ.
Professor Caroline joined or not? I don't know. Let me check. There are no came, no?
No. No, sir. No, sir.
Yeah, I think she has joined. Madam is joined. No, request to join, we can call them.
ओपन है वो सर है।
Welcome, Professor Korone. Siva Kumar here.
Hello, welcome. Thank you very
Waiting.
much for the
Yeah.
invitation.
Yes, sure. So we will start in 10 minutes, 3 o'clock exactly. Okay.
Yes.
Thank you.
I can maybe already try to share the screen.
Yes, please.
Okay. You can see it in full?
Yeah, it's good.
Great. Wonderful.
Thank
Okay.
हाँ तापसी आज्ञा लैब कोठी आमे तो एठी अच्छो सबू आ तमे जीव जदि जाओ किछ असुविधा नाही 5टा 6टा हे जो माने 4:40 टाइम अछि किंतु आनी के सब याडे सेटअप करियर अछि तो सेथि टाइम लागि परे हो हम्म हम्म ये बहुत गरम लाଗୁଛି ସେଟା ଏସି ଟା ଅନ୍ ଅଛି ତଥାପି ଗରମ ଲାଗୁଛି କାଲି ଏଇଠି ବି କଲେ ମ୍ୟାମ୍ ଚଳିବ ମତେ ଲାଗୁଛି କାଇଁ ଏଇଠି ବି କଲେ ଚଳିବ ଏସି ଚାଲିବନି ପରା ଓହୋ ହଁ ସॅଟରଡେ କେତେବେଳେ ଚାଲିଛି କେତେବେଳେ ଚାଲିବ ସେମିନାର ଏସି ଅଛି हेलो मैम आज्ञा मैम हाँ 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 आसले स्टार्ट माने एबे हबो हेइनी एबे स्टार्ट हो हाँ आज्ञा मैम उषा मैम भी आसी जाइछन्ती त ठीक अछि आज्ञा
You start recording.
हाँ yes sir so ma'am called
She has not come.
she will come like it means time लगेगा ऐसे तो
Time लगेगा.
means introduction you otherwise उषा मैम will
अरे उसका practice नहीं किया कैसे करेंगे?
she now only she called recording start ना हाँ
Start. वो YouTube channel नहीं कर रहे.
हाँ started
कर
that
दिया.
one also continue here that is going on
इसका अभी कौन पढ़ेगा उसका?
मैम
Good afternoon, one and all. It's very great pleasure to invite Professor Caroline for this exciting world of organic electronics and for this international webinar. Thank you, Professor, for accepting our invitation and then giving lecture for our community. And it's my privilege to read Professor Caroline biography. Dr. Caroline received PhD in physics from University of Technology Dresden, Germany in 2015, working on excitonic processes in OLEDs under the guidance of Professor Carl Leo. Subsequently, she performed her postdoctoral research under Marie Curie Fellowship in the group of Professor Malte at the University of St. Andrews. After completing her postdoctoral research, she joined Kurt Schabbe Institute for Measuring and Sensor Technology in Menzberg, Germany, as a junior research group leader in 2018, where currently she is working on new applications of organic semiconductors in biophotonics and medicine. Professor Caroline, please take.
Thank you very much for this kind introduction. And it's my great pleasure to present you our research today, which will be mainly focused on the applications and on how we can drive OLEDs towards these specific applications. So neuronal interfaces is maybe something that not everyone is familiar here. So I want to give a brief introduction on neurological diseases. Neurological diseases affect more than 1 billion people worldwide. You all know the most common ones are Alzheimer's disease, epilepsy, but also migraine, for example, and many other neurological diseases from which people can suffer a lot. And in order to better understand these diseases and in order to cure them, we need to have methods in order to control nerves. And the methods that are used so far for this are mainly pharmacology, so delivering drugs, which is not at all timely. It's also not well targeted and comes usually with many side effects. And the alternative for this that is most often used is electrical stimulation, where basically little electrodes are placed close to the targeted cells and then can deliver little electrical impulses. So this is, for example, shown here in such an epilepsy recognition and treatment device that exists already. So basically this can be implanted with little electrodes that can detect an epileptic seizure and then send out an electric impulse in order to suppress this seizure. The electrical stimulation can be very timely, but the problem is that it is not targeted to specific cell types. Instead, it's basically stimulating all cells that are in the vicinity of the electrode. And it also does not allow silencing of cells. So instead of silencing the overactivity during an epileptic seizure, one sends out an additional activation pulse. So researchers were eagerly looking for methods in order to increase the targeting and specificity and also enable silencing of neurological signals. And the method that they've come up with in 2005 is called optogenetics. And this uses light in order to control cellular function and behavior. This is done in an artificial way by genetic modification, and it works like this. We have here a very nice illustration of this, a cell basically getting a light pulse and then in response to this starting to fire. And the working principle is as follows. In the neuron, in the cell membrane of the neurons, there are these tiny little pores that regulate the flux of ions through the cell membrane. And these ions can now be made these ion channels can now be made light sensitive through genetic modification. which means that whenever we shine light onto this ion channel, we can regulate either an opening or also a closing of this channel, and therefore regulate the flux of ions through the cell membrane. And this means that on one side we can bring neurons to fire. In this case, we would have cations flowing into the cell. Or we can also silence neurons, for example, by pumping cations out of the cell or by leaving anions into the cell. This method is very powerful, has led to many developments and breakthroughs already in neuroscience especially, and it is also now investigated in some first approaches in order to apply this even in humans. One example for this is the optical cochlear implant. So maybe you know about the electrical cochlear implant, which can help deaf people to gain some hearing again. So this means one basically introduces a number of electrodes into the cochlea that can then stimulate or send electrical pulses that have been recorded with a small microphone. And with this, it is possible for deaf people to at least follow a conversation in a quiet room. But the problem with, as I said before, with the electrical impulses is that their stimulation is very broad. So it spreads over many micrometers, up to millimeters, the signal, and therefore it's hard for them to distinguish between the low and the high frequencies that we can hear. And the optical cochlear implant instead can focus light much more into only very narrow frequency regime. So the idea is that with an optical cochlear implant, it may be possible in future to restore hearing functionality with a much greater precision than what is possible with the electrical implants. And I want to touch just one other example. Optogenetics is also suggested to be used for curing specific forms of blindness called retinitis pigmentosa, where basically the receptor cells here are made light sensitive through optogenetics, so that they can sense again some light intensity. The only problem is so far that these optogenetic constructs are usually not as light sensitive as our natural light sensitivity would be, and therefore one requires a specific goggles that have a camera which can take up the ambient light, and then a projector with a high power light source that can project the image onto the retina. And this is actually already in clinical trials. There has been some success already in human patients, so it's really a very promising method. The light sources that are used for optogenetics so far, then the most prominent light sources are optical fibers that in a more advanced fashion can also come in these fiber bundles, so one can get a little bit of spatial resolution. Still pretty much limited, and of course the optical fibers are rigid. It's not very convenient to attach them to a brain in a mouse, for example. And alternatives for this that have now come up are flexible micro LEDs, as have been used, for example, in such an optical cochlear implant. But if we look at the overall structure, we have here approximately 1 millimeter bending radius. The whole system is 300 micrometers thick, and of course LEDs themselves are usually not flexible, so one can just create such demonstrators by picking individual LEDs and placing them at a relatively large distance in order to gain some flexibility. And here our is to use instead OLEDs, and we've already heard this morning a number of advantages why OLEDs can be helpful, but I want to go specifically here into why OLEDs are useful for optogenetics. And so first of all, as we already heard in Professor Adachi's talk this morning, OLEDs enable structuring to very high density arrays, also with losing less efficiency compared to inorganic LEDs if we scale them to small areas. You can see this nice example here of a microscopic OLED array where each pixel is 6 by 9 micrometers size. This is smaller than the size of a neuron. Of course, OLEDs allow a very high spectral control, and this will be important in order to match the emission spectrum with the activation spectrum of the used light-sensitive proteins. And of course, OLEDs can be made highly flexible, not just in with 1 millimeter bending radius, but even much more flexible because they can also be made much thinner. And an additional advantage of OLEDs is that it is also possible to emit multiple colors from exactly the same spot. So this example here shows a stacked device with red, green, and blue emission, each operated individually on top of each other, which enables to use the same pixel but for multiple color stimulation. And I will come back to this in my research later in the presentation. Okay, so I hope you see that OLEDs can be very helpful in this regard, and also for other biomedical applications, which also have been investigated, like for health monitoring, for therapy, or lab on a chip. But I want to focus onto optogenetics and brain interfaces for today. But there are a number of challenges involved and specific requirements that need to be met in order for OLEDs to be useful for optogenetics. So first of all, we need to have a very good spectral overlap of the activation spectrum of the used light-sensitive protein. These are often called channel rhodopsins. That's what the CHR stands for. And of course, this needs to overlap well with the OLED emission spectrum. Second, we want to have only low temperature spreading onto adjacent cells, because cells need to be kept at a specific temperature and we can't grill them at very high temperatures, so then we would kill them. So this refers to pretty good device performance. We need to have devices that are highly durable in an aqueous environment. So we need a very strong encapsulation in order to protect the organic layers from oxygen and moisture. We also want to pattern the devices to pixels in the size of cells, so at around tens of micrometers. And depending a little bit on the application and on the actual system used, we may also need very high light intensities in the range of around 0.1 to 10 milliwatt per square millimeter. This is usually what is considered necessary for optogenetics. So in my talk, I want to focus onto these three different aspects of OLED requirements for optogenetics, and I will start with the device performance. In our work, we have predominantly concentrated on using fluorescent OLEDs for some reasons. You can see the stack that we use most often with slight variations in our work. It comprises the fluorescent emitter TBPE. And the reason for using fluorescent emitters is that we want to drive the devices at extremely high light intensities, where the phosphorescent or TADF OLEDs would suffer strong efficiency roll-off. And on the other hand, fluorescent emitters usually allow to drive the devices at lower voltages. And this is also quite important in order to get high luminance at low voltages. This is required, for example, if we want to incorporate OLEDs later on silicon CMOS spec driver electronics, then these are often limited to 5 volt. So this means we want to have as high luminance at 5 volt as possible. So, and for this reason, we also use doped charge transport layers here. So we have a p-dopant F6-TC-NMQ, and we have an n-dopant cesium here in order to have a very good charge injection from the electrodes, and also in order to have no voltage drop across these two transport layers, but instead having the voltage drop only over these intrinsic layers. We did some variations on the stack. So for example, one example is shown here, a variation of the electron blocking layer. So we used these 5 different materials here, NPB, TAPC, alpha NPD, Spirotut, and BFDPB. And we studied, on one side, we wanted to have very high current densities. And we studied this also as a function of the thickness of the emission layer, I believe. Sorry, I think there's a mistake here. So of the emission layer, where we see, of course, thinner emission layers, we can get to higher current densities. But if we look at the EQE at 5 volt for these different devices, then we also see that the thinner emission layers will have a drop in EQE because some of the emission gets quenched by the adjacent blockers. We also looked at the lifetime of these devices, and both in electrical and EQE properties, and also in the lifetime, Spiro-TAD was the best candidate for us. And I want to show you the results of this of these OLEDs. On one side, as I said previously, we need to have a spectral match of the device with our activation spectrum. So the black line here is the activation spectrum of such a typical light-sensitive protein. And you see that with our blue OLED, we match this activation spectrum quite well. And on the other hand, I told you that we need high current density and especially high power density output at low voltages. And the OLED here with the Spiro-TAD reached approximately 1 amp per square centimeter at only 4 volts. It's pretty high current density. And if we look at the optical power density, we could exceed 2 milliwatt per square millimeter at 5 volts. We also studied the lifetime of these devices in a pulsed mode, because later we would mainly use a pulsed driving scheme in order to stimulate cells for some milliseconds. So in this case, we have here 10 milliseconds and 10 hertz driving. And we drove the device at an initial brightness of, or power density of 0.5 milliwatt per square millimeter. And this is much, much higher than in what you have in display. So maybe because if we convert this to candelas per square meter, I believe we are in the range of maybe 60,000 candelas per square meter or so for blue OLED. So this is much brighter than what you would use for displays or so. So for displays, we are more in the range of microwatt per square millimeter. Yeah, and over this driving course of 1 million pulses, we have a decrease of OLED brightness of around 20%, which I think is acceptable, and which is also at least supporting the fact of using fluorescent emitters, especially for the blue. I believe with phosphorescent and TADF emitters, this brightness drop would be significantly stronger. Okay, in the next step, we applied these OLEDs in a different neurological model system, and I was particularly working a lot with Drosophila melanogaster. That is a common fruit fly or vinegar fly, and we were not using the flies, but mainly this dirt instar larva from the flies. And we used larvae that were expressing light-sensitive proteins in sensory neurons that are basically sitting all above the larva, which means that when we shine light onto the animal, this will lead to a muscle contraction. So the larva will sense this activity, the muscles will contract, and if we inhibit this cell, so if we turn off this sensory cells, then it will lead to a slowdown of the muscle contraction waves. So in our studies, we used 2 different light-sensitive proteins. One is called crimson. This is particularly sensitive in the red and can be used for activating cells. And we used another one called GTACR2, which is mainly responsive in the blue wavelength regime. And with this, we can inhibit cellular activity. So what we studied first is the OLED power density that we needed in order to elicit neurological response for these different proteins. So in case of the activation with the crimson, we achieved this in these larvae already starting from 2 microwatt per square millimeter. So they are actually very, very light sensitive. And the inhibition was possible from approximately 10 microwatt per square millimeter onwards. It is also possible to not only have individual OLEDs and then do either an activation or an inhibition, but pretty recently collaborators of us have developed a new neurological tool called bipoles, and this is a fused protein, so it connects the inhibitory part, the GTACR2 that I was talking previously of, together with the crimson into one compound, which means shining red light onto the cell will lead to activating the neurons, and shining blue light onto the neurons will lead to inhibiting neuronal activity. And we thought OLEDs are the perfect light source actually to do this, because we can stack them on top of each other in order to provide emission from exactly the same spot, but with 2 different colors. And for this reason, we've developed these bicolor called AC/DC OLEDs. The actual principle is already some years older, was developed at TU Dresden. The idea is that we have a phosphorescent red OLED on the bottom and a fluorescent blue OLED in the top. So that's a top emitting device in total. And we put the outer 2, this one and this one, onto the same potential. So this means we can either drive this unit by applying positive voltage here, negative voltage here, or if we reverse the bias, have positive voltage here, negative voltage here, then we can light up the blue device. And we can basically switch through this blue or red emission in a pretty fast manner. You see that the overall stack is pretty complicated, and especially in top emitting OLEDs, it's very important to consider also the optics of the device. And therefore, we simulated the emission spectra, varying the thickness of the electron transport layer in the red device and the hole transport layer in the blue device. And what we can see as a result is that when changing the electron transport layer thickness, so by increasing the thickness in this red stack, we get a significant shift of the red emission spectrum. And if we change the hole transport layer thickness here, we get some change in the emission spectrum of the blue device. So this is here at 25 nanometers, and this is at 65 nanometers. This is significantly green shifted here. And our goal was to match or to basically separate the blue and the red emission as much as possible, while still having a good overlap with the activation spectra of these 2 different proteins. So these are shown here in blue and in red in the background. So in our final device, we used 25 nanometers hole transport layer for the blue and 55 nanometers, so this yellow curve here, in order to have a good spectral separation, so that we will not stimulate the blue channel by the red device or the red channel by the blue device. So in our actual devices, the spectrum was matched quite well compared to the simulation. So we have simulation now here in orange, and the red and the blue are the experimental spectra of the devices. And if we now look at the performance of these devices, this is shown here in this graph. We have the black lines showing the current density, and you see now that when applying forward bias, we have red emission, and if we apply negative bias, we have blue emission. The irradiance is somewhat lower in these devices, so it's reaching approximately 100 microwatt per square millimeter. Main reason here is that these devices were pretty large area, I believe around 18 by 18 millimeter square. We also studied the external quantum efficiency, both in simulation and in experiment, and this is shown here. So again, for different thickness of the electron transport layer, we will also vary the external quantum efficiency, of course, because we can have higher or lower outcoupling efficiency. This is particularly visible here. In this case for the red stack, where we can change the external quantum efficiency between around 3 and 14%, depending just on the thickness of the electron transport layer. So in the devices that we fabricated here with these thicknesses, we achieved around 12% EQE in the red, and the blue is up to around 3% EQE. Remember that this is a fluorescent emitter, therefore can't expect too much from this. Yeah, as I said previously, it's also important to look at the temperature development of the devices, and we measured this for a sequence of positive and negative bias, so either red or blue emission of the device, where the device was driven with 5-second pulses alternatingly, and we had an increase in the temperature of around 2 degrees during this time. That's still a little bit too much if we want to get in close contact with cells, but this could be improved when improving the device performance, and especially also using different driving schemes, because this is really like turning or keeping the devices on for like 250 seconds. We now use these devices in 2 different optogenetics experiments. One is in cells, where we controlled the photocurrent that was basically going through the cell membrane. So with the blue light, we now had a high depolarization of the cell, and with the red light it was then the inverse. So we could basically flip between having incoming currents or currents going out of the cell, and this as a function of the irradiance of the device. We also use these devices for stimulating, in this case, motor neurons in Drosophila larvae. So what you can see here is a larva on top of an OLED, and when we turn on red light, then the larva contracts, and when we turn on blue light, the larva relaxes and therefore elongates. And we've done this in a multiple way of stimulation and inhibition of the neurons, and you can see here the length of the animal plotted over time, and you can see clearly you have contraction, relaxation, and so on. And I also want to show you one video on this. I hope that you can see this with the connection. So what we have here is multiple larvae on top of our OLED. And when we now turn on the OLED, red, blue, you can hopefully see that the larvae shrink and elongate. Okay, so far I've always just showed you examples of using OLEDs that have a pretty large area, but we are also looking at patterning the devices to smaller area in order to stimulate cells with higher specificity. And for this, we've structured OLEDs basically by patterning the bottom electrode into such a one-dimensional array. So each of these little stripes can have a width between 10 and 100 micrometer, depending on the system that we use. And in order to also get a higher resolution, when we bring this device in close contact to cells, we need to have a very small distance between the OLED and the animal, for example. And in order to protect the device, we used 30 micrometer flexible glass encapsulation that was attached directly on top of the OLED. The performance of these microstructured OLEDs are comparable to the large devices. So you see here the current density and the EQE of the devices. There's no significant change by jet patterning of the bottom electrode. Yeah, we applied this again for our Drosophila larvae, and what you can see now here is our OLED display underneath the microscope, and we have here a larva. So to give you an idea, this is larva is approximately 4 millimeters in length, and we have here the head and here the tail. And what happens if we now stimulate this larva in different places is that we can either elicit such a contraction wave, or we can also hold a muscle contraction wave at specific, by stimulating specific neurons in the body of the animal. And we use this for quite some detailed neuroscience studies. I don't want to bore you with these results. I just want to show you one more video of what can be done with this. That again, the larva head is on the left and tail is on the right. And in the moment the larva is crawling backwards. And when we stimulate it in the center or in the back of the animal, then the larva switches its crawling direction and now crawls forwards. And if we now stimulate in the front of the animal, we can again switch the crawling direction and it's now crawling backwards again. So we can basically use OLEDs in order to control into which direction the animal is moving. Optogenetics is very useful for stimulation of cells, but there is another technique which also enables sensing of neural activity, also by using light. And this is by using genetically encoded fluorescent indicators, and most prominent are so-called calcium indicators. So these are basically fluorescent proteins, and upon binding of calcium, they undergo a conformational change and therefore will change the photoluminescence quantum yield of the fluorophore, and therefore by exciting these fluorophores and reading out the emission intensity, one can read out the activity of the neurons. So this means if we look back at this image here, we cannot only stimulate the cells with a specific color, but when expressing such light sensitive or light emitting fluorescent indicators, the cells can also emit light, and in order to do so we need to have another excitation light source. So in this case this would, for example, be green, so that the fluorophore will absorb the green light and then can re-emit in the red. And this is also a very common technique that is used in neuroscience in order to create maps of neuronal activity, as shown, for example, here. But the problem is that so far, usually the animals are constrained underneath a microscope, so it cannot move freely, and this is pretty much limiting on what we can actually learn from the behavior. So the idea is that it would, of course, be much better if we have a small implant, flexible with patterned structure, which can be used in order to read out neuronal activity. And as I said, we need a light source in order to excite these fluorophores, and this is what we've been looking at using, again, OLEDs. So in order to design OLEDs for this purpose, we first need to have a look at the excitation spectrum of the typical light-sensitive proteins, and we specifically focused on the green fluorescent protein shown here, which is one of the most common ones. And you see here the excitation spectrum and the emission spectrum of this fluorescent protein. So usually one would use a blue laser or so, excite here, and then filter out just the emission between 500 and 550 nanometers to look at the emission from the fluorophore. Now, we want to apply OLEDs for this purpose, and we've used, again, similar structure like what I showed you before already. But if we look at the OLED electroluminescence spectrum, and this is now shown on a log plot here, so this spectrum looks extremely broad in this case, we see that we have a very good overlap with the excitation spectrum, so we can have pretty good excitation, but we also have very strong overlap of the OLED emission in the wavelength regime where we only want to see the emission from the fluorophore. So we basically need to get rid of this area of this emission of the OLED in the green wavelength regime. And if we look at how much we need to get rid of it, we made a small calculation, which is shown in this graph here. This gives the number of photons that are absorbed and re-emitted by the fluorophore as a function of the concentration of the fluorophore. And if we look at the typical concentration range in cells shown here, then we are approximately at a factor of 1000, meaning that only every 1000th photon will be absorbed and re-emitted. And therefore, this part of the spectrum basically also needs to be suppressed by an order of, by approximately 3 orders of magnitude. So in order to get rid of the emission in this wavelength regime, we used distributed Bragg reflectors. So basically a Bragg mirror that consists of alternating layers of 2 materials that have a different refractive index. In this case was tantalum pentoxide and silicon dioxide. And we attached this filter on top of our OLED. And as a result, you can see here we can really cut off the emission in this wavelength regime entirely and mainly have the blue emission here. But this is a spectrum at 0 degree, and I think many of you will know that if we look at different angles here in such a Bragg reflector, we get a significant shift of the transmission wavelength. So if we look at higher angles, then starting from approximately 40 degree onwards, we will get again significant emission from the OLED in the area, in the wavelength regime where we only want to see the emission from the fluorophore. So this is limiting our devices in the moment to approximately 40 degree angle from which we can collect light. This is actually important if we look at the devices with a microscope. Then we have an objective with a numerical aperture, and the larger the numerical aperture, the larger also the collection angles. But nevertheless, we used this device and put it underneath the microscope and stimulated different samples with it and looked at it here with the objective from the top. And in result, we saw that we can basically achieve fluorescence imaging with the OLED at a similar quality as with conventional mercury light sources. And we also wanted to do neuronal imaging, and this is shown here. What you can see here is the central nervous system of a Drosophila fly, and stimulated with an OLED light source, and we look at the emission of the fluorophore, and we could see such a waveform activity going on in the brain, which gives us basically a proof that we could successfully do calcium imaging using OLEDs. In the future, we aim to combine this optical stimulation with OLEDs with sensing. We did some first studies in this regard using an OLED as an excitation source for exciting cells and doing fluorescence imaging at the same time, but not using OLEDs, instead using conventional LED light source. And we did this in some cultured hippocampal neurons. I won't go into much detail, but what we could see is that after stimulating the neurons with some light pulses, we could see a significant increase in the fluorescence intensity. That's also shown here in these images. After stimulation, we have much brighter intensity than before, which proves us that the OLED could elicit neuronal action potentials. In my last slide, I also want to show you our efforts towards providing flexible devices. And I think you probably all know that it's very important to keep OLEDs, protect OLEDs from water or oxygen in the environment. And especially if we want to use the devices in contact with cells, then they really need to be waterproof or proof to the physiological solutions. And for this, we've developed a new substrate and encapsulation that consists of alternating layers of the polymer parylene and a so-called nanolaminate. And the nanolaminate is fabricated by atomic layer deposition, which is a method that enables nanometer thin layers, but with extremely high conformability, so they can penetrate extremely deeply into voids and therefore offer very good protection. So we studied these alternating structures of parylene and nanolaminate, where each of these is a 6 micrometer with our OLED in between. So we have samples that are in total approximately 12 micrometers. And you can see that they are really highly flexible. This is bent around the backside of a razor blade. And we can also operate them actually in water. And this is shown in this video here. So that's an OLED operated in DI water. And I hope you can also see that it is really a very flexible device. And it's also not only working for this one-time video, but we also investigated the OLED over the course of 2 weeks. And what we could see is that over 2 weeks, and using this structure with parylene and nanolaminate, we could keep the luminance constant over 2 weeks. So we had basically no degradation of the devices in water or in phosphate buffered solutions or cell culture medium. Okay. So with this, I'm at the end of my talk. I showed you our different investigations towards the development of OLEDs, how we could employ it for optogenetic stimulation and also for fluorescence imaging. And I believe and hope that we can at some point create such a really high resolution all optical systems, so having OLEDs for optogenetic stimulation and for fluorescence imaging, and maybe also combine this with readout systems later to have create such implants that could be used for neuroscientists to better understand our brain, or also for functional implants. With this, I thank you very much for your kind attention. Also, I thank very much to the organizers for inviting me. Thanks to my group at Kurt Schwabe Institute, and also to our collaborators. Okay, thank you.
Thank you, Professor Caroline. Now we have a couple of quick questions. If anyone asks, please raise your hand, or you can. We have time, no problem. Professor, what do you think about this NIR OLED for this biological application? Because morning, like many of them, I mean, three of them almost touch the NIR OLED. So
yeah, I think there is definitely great potential for NIR OLEDs in biomedical applications. And I think it's very important to move on towards developing more efficient emitters for near infrared. So yeah, I think that there would be good applications, especially also for sensing. I mean, there's also in terms of optical sensing, there's lots of development going into near-infrared photodiodes, organic photodiodes, and if these could be combined with OLEDs as a light source, I think this would be a really great achievement. So far, I think it's mainly lacking on the OLED side.
You have any future direction in that line in your group?
Not with NIR, but we want to combine organic photodiodes for sensing neuronal activity. Yeah, that's our aim for the next years.
That's great. Okay, Professor, thank you so much for joining, and we look forward to some
thank you very much.
In person in India. Let us hope for the best. I mean,
this would be great. Or in Germany. Thank you.
Thank you so much, Professor. Welcome, Professor Data. This is Siva here.
Hello, do you hear me? Do you see me?
Yes, we are hearing.
Okay, that's great.
Yeah. Shall we go ahead, Professor? Or
do you see my slides?
Yes, it is visible to us.
Great.
So, shall we start, Professor?
Yes, I think we can start.
Go ahead, yeah. Okay. So it's my privilege again this first day of our webinar and the second talk in the session 2. We have with us Professor Data from Chilchan University of Technology, Poland. Now I request my colleague Professor Usharani Subudhi, co-convener of this event, will introduce Professor Data.
Thank you, Professor Siva, for this opportunity. So it's actually a proud privilege to introduce Professor Data. Professor Data is currently a professor at the Silesian University of Technology. He is also the director of the Center for Organic and Nanohybrid Electronics at the university. He started his scientific work during master studies, which resulted in employment at the Center of Polymer and Carbon Materials in the year of 2007, Polish Academy of Sciences. After that, he earned his master's in Science and Technology and PhD in the year of 2013 from the university, working with Professor Lepkowski. He pioneered the use of selenophene and tellurofene-based conjugated polymers as both emitters and electrochromic windows in devices. After completing his PhD, Dr. Data received 2-year postdoctoral fellowship funded by Polish Ministry of Science and Education and worked on the use of exciplexes as TADF emitters in organic light emitting diodes. With Professor Andrew Monkem. He is also a recipient of Marie Curie Individual Fellowship. After the fellowship, he was appointed as the experienced researcher in Durham University jointly with Silesian University of Technology and continued to work on organic electronics. Professor Data has many prestigious awards to his credit. He has achieved Polish Prime Minister Award for Outstanding Doctoral Dissertation, Individual Award of Rector of the Silesian University of Technology for Scientific Achievements. He has also received the 2017 edition of MIT Innovator Under 35 Award and MSCA Individual Fellowship, to name a few. So we are really happy to have you here, Professor Data, and over to you. And we are looking forward to listen to you.
Thank you very much. First of all, I would like to thank organizers for invitation to be able to speak here and to show some of my work. So to be honest, I'm chemist, mostly my background is electrochemistry, spectroelectrochemistry of organic compounds. Nevertheless, I work more than 5 years in Durham University in UK, working in the physics department, mostly on the photophysics of organic materials and also on the OLED devices. So nowadays, I would say I'm more material scientist, but because of my work in both physics and chemistry, I learn how to gather all the informations and use them for explaining different phenomena. And here I would like to show today's couple examples of the analysis of OLED emitters, especially TADF emitters. So first of all, where I am, I'm in Poland. And as I understand, the organizers of this conference are in Rourkela, India. So you see that on the slide it's not so far, but actually it's a couple thousand kilometers far from the university. So Poland is here, and if we think about Poland, which is a much smaller country than India, our capital is Warsaw in the middle of Poland, but we are in Gliwice here, a little bit smaller town near Krakow. Maybe you already know Krakow. It's a very beautiful old town, and it's around 100 kilometers from it. So whenever you will be in Poland, in Krakow or Warsaw, I'm inviting you to come here and talk and see what we are doing. And that's our quite old department where we are conducting not so old research about OLEDs. So what we can say about OLEDs, organic light emitting diodes? I will start from basic because it's important for further explanation. How they work. Here is an example of one of the simplest efficient structures. So we have our anode, which is usually indium tin oxide layer, which is transparent or semi-transparent layer, and it's a source of holes for our devices. On the other side, we have cathode, usually aluminium. There are other possibilities, but aluminum is one of the easiest to use. And we have electrons which are injected into our devices. In meantime, in the middle, we have different organic layers, hole transporting layer, emissive layer, and electron transporting layer. From the basics of the organic semiconductors, you should know that we have a valence band and the conductive band. And we have two types of conductivity or conductance. We have hole conductance and the electron conductance, conductivity. So if you are also organic chemist or quantum mechanist, if you describe organic molecules, you always think about HOMO and LUMO of your organic compound. So here, if you think about that, this box is organic compound. So on the bottom is the energy level of your HOMO, highest occupied molecular orbital, and on the upper side is the level of energy level of your LUMO, lowest unoccupied molecular orbital. So we have our valence bands, and here our holes are going through our organic compounds up to emissive layer. On the other side, the same with electrons. They are going through the conductivity band. Our electrons are moving through the LUMO level up to our emissive layer. And we want to have both holes and the electrons in the emissive layer. Why? Because they form excitons here. Excitons are the I would say artificial form where we are binding our holes and electrons forming exciton, and when they are merged together, they emit energy in the form of light. So that's the simplest way explaining how we are getting from our holes and electrons our emission in the organic compound. So that's about the for the OLED structure. But if you'll go through our emitter, because we want to synthesize, improve, or generally find our efficient emitter. In general, in organic emitters, when we excite our molecule, it doesn't matter if that's electrical or photo excitation, we are forming excited state. But it's not so simple that we are forming excited states. We are forming two different excited states. One is the singlet excited state, and the second is the triplet excited state. And to complicate that a little bit more, from the mechanism point of view, for one singlet excited state, we are forming always three triplet excited states because of the spin rotations in our molecule. Why it's important? Because when we excite our molecule, for example, we add energy. If we have only fluorescence emitter, that means we can harvest only 25% of our energy which we put to our device or material. So it doesn't matter how much you will put in the beginning energy, how much you excite your molecule, you are getting only 25% of your energy in the form of the fluorescence, so in the form of the emission. In the other case, where we have three triplets, it's a little bit better because we can get here 75%. So we have here 75% of excited state. But of course, in the devices, especially in OLEDs, we want to have as high efficiency as possible. So we want to use all of the efficiency, 100% of energy which we put into. To do that, we have, as scientists develop phosphorescence emitters, metal organic emitters, where they have inside phenomena called metal ligand charge transfer. I won't focus on that more. I only telling that there is such effect. You can read more about it, but generally how this is helpful here, it allowed us for a very fast intersystem crossing from the singlet to triplet state. Why? Oh, I forgot to mention. In typical, when we have fluorescence process, fluorescence is the nanosecond emission. So emission is very fast. In other case, where we have phosphorescence, as you probably already know, phosphorescence is a long-lived process. Why is long-lived? Because our spin, which need to flip, and it need to move through the intersystem crossing, we could say like that, and it need time. It need time to cross, and that's why phosphorescence is long-lived. Nevertheless, in the organometallic complexes, thanks to this metal ligand charge transfer process, this process is much faster. So where typically phosphorescence can go millis or even seconds lifetime, in the organometallic phosphorescence emitters, the lifetime drops to microsecond time. And that's important for application in OLEDs, especially if you think about your mobile phones, AMOLED screens. If you have in the lifetime in the milliseconds or in the seconds, you will need to wait till your face or your video will unfreeze on your screen, and we don't want that. So our emission should be very fast. And for that, phosphorescence emitters, metal organic emitters were quite fruitful. Nevertheless, most of the phosphorescence emitters are organometallics, which contain iridium complexes or platinum complexes, and they are quite expensive and not so easy to get. Because of that, scientists start thinking to use another process where only pure organic molecules could be used, or non-heavy metal containing molecules. And this process is called delayed fluorescence. And what's the behavior or what's going on inside? We are going in the completely opposite side to the phosphorescence emitter or metal complexes, and we are trying to harvest our triplet state. So our 75% of the energy is in the form of the excited triplet state. We are pushing to the singlet state. So the singlet state where we have fluorescence emission. But because again we need to have a spin flip and we need to go through the firstly intersystem crossing and then reverse intersystem crossing, it takes time, and that's why we don't have normal fluorescence but the delayed fluorescence process. Nevertheless, in both of the cases, phosphorescence emitters or delayed fluorescence emitters, we can harvest up to 100% of our excited states. And that's our goal in the new OLED emitters, to use delayed fluorescence and get as high efficiency as possible. Nevertheless, like everything in the life, it's not so simple, because we have two types of delayed fluorescence process. One is called E-type. E-type delayed fluorescence is from eosine, when it was firstly discovered, and the second is the P-type. P-type from pyrene, where it was also discovered. So what is the important factor here? Why there is a difference between those two delayed fluorescence? In the first case, our gap between triplet and singlet state needs to be as small as possible. They must be as close to each other as possible. And from the experience and literature, you can find that to have efficient transfer from the triplet to the singlet state, the gap should be lower than 0.4 electronvolt. Why 0.4 electronvolt? Because in general, this is the energy which you can harvest at room temperature from the heat. And that's why this process is also called TADF, which means thermally activated delayed fluorescence. The other process where the gap is higher, we have another process, which is called triplet annihilation, TTA process, where we need to use two triplets which annihilate and form one singlet excited state. In the TADF, we are harvesting 100% of the triplet state, which those excitatories are pushed to the singlet. And in this case, in the case of TTA or P-type delayed fluorescence, we can only harvest 50% of our triplet state. So if you think about 25 to 75 ratio, in the case of the P-type, we are harvesting only 50% of the 70% of excited state, which means in all the cases we are pushing for TADF process run than TTA. Nevertheless, in most of the in many molecules, it's mixed of the processes E-type, P-type, and that's why we always need to investigate that. So we know what the mechanisms are, but we need to investigate. We need to actually know if our molecule have a E-type or P-type, and if it's possible, we need to screen that to know if it's possible to use that for OLED, if there is a reason or chance to get efficient OLED. And how to do that? Usually we are using very nice equation. If you think about your organic molecules, if you are screening of them, you are measuring your emission. So you are always analyzing your PLQI, so photoluminescence quantum yield. So the amount of photons emitted, if you see here, to those absorbed. If you think about devices, OLED devices, our parameter is external quantum efficiency. Which means how many photons we generated and how many electrons we push into our devices. And there is a really nice equation which previously Ken showed us, where we can combine our EQE and our PLQI of our emissions. So we use this equation to screen our materials quite fast to know if our organic emitter will be efficient in the form of the OLEDs. As one of the important factors which you need to know here is this fraction of the excited state. So in the case of typical fluorescence emitter, our fraction is 25%, so 0.25. If we think about TADF, it can go for 1, so 100%. If we'll compare those different processes, emissive processes, you can find that for normal emitters, fluorescence emitter, our EQE can go up to 5% if we assume that our outcoupling factor is 0.2. Again, in normal typical devices without any boosting, optical boosting, the outcoupling is between 0.2, so 20% and 30%. And what is outcoupling factor? If you compare like OLED device and very old light bulb, it's the amount of light which is going directly to you. You have flat surface, the light distribution is different. You have different angles of your light coming to you. And in the case of devices, this is the value which is of the light which is going directly to you. Higher value, the light is more focused into one direction. That's the simplest explanation. And the typical devices, the value is around 0.2. That's why we assume in all of them the outcoupling is 0.2, and then depending on the mechanism, if we have fluorescence, we can get 5% of the external quantum efficiency of the OLED device. If we have only TTA process, we can go up to 12%. And in the TADF or phosphorescence or room temperature phosphorescence, which is also the case, we can go up to 20%. In the literature, you can find devices where they have like 30% or 40%, and that's only because you are changing the outcoupling factor. I forgot about that. So analysis. How to analyze your organic compound to use as the OLED emitters, TADF emitters. First, in our case, we are doing the electrochemical analysis, which is one of the simplest. You have cyclic voltamperogram, cyclic voltamperometry. Why? Because of the two reasons. Firstly, to check if our compound is ambipolar. So if we can put there, we can inject both electrons and holes, because that's important for the devices. And also to know what is the HOMO and LUMO levels, because this factor will tell us what the supporting, like the whole transporting layer or the electron transporting layers we can implement in our devices. So those values are important for the tailoring on the final device. And we also, thanks to that, we also know if our compound is stable. So if our oxidation or the electron doping is a reversible process or not. If it's not reversible, then we need to find what's going on, why it's not reversible. So that's why we are starting, for example, in our case, from the electrochemical analysis. The other factor is a simple emission spectroscopy. So we are checking how our compound is emissive, how strong. So we can also do the PLQI, like I said before. But what is more important in the case of the typical CT-based EADF emitters, we want to know if there is actual CT state, because we have donor and acceptor molecule, and the CT, like in the phosphorescence, is boosting or is increasing our speed of the transfer from the reverse intersystem crossing process. And how to check using typical fluorescence spectroscopy? Simple, by checking how the molecule behave in the polar and non-polar solvent. In the non-polar solvent, we should have very nice, very narrow emission, which is usually associated with the localized excited state. But if you will move to the polar solvent, like here in the red, then we have very broad emission, single exponential emission, which is associated with charge transfer process. So this is the charge transfer state. In some of the cases, if you look like the middle one, we don't have emission in the THF tetrahydrofuran at all. What is important for you to know, it doesn't mean that we don't have charge transfer process or there is no charge transfer in the molecule. Sometimes the emission disappeared, mostly because the charge transfer process is so strong that a very small amount of oxygen simply dissipate quench your whole of your emission. If you would have emission in THF, which is almost exactly like the one in cyclohexane or in non-polar solvent, that means you don't have CT emission at all, or CT charge transfer process in your molecule. But if it disappeared, that means there is a chance that your emission is quite strong. Then we are going to the more sophisticated analysis, time-dependent spectroscopy, time-dependent emission, where we are using our laser setup and we are exciting our molecule in the nanosecond times, and then we are checking, we are gathering the emission spectra at different times from the nanoseconds up to second time region. So you have here a transient curve, but those curves actually are represented by spectra. At every point of the curve, we are obtaining spectra. So we can see how our spectra is changing at different temperature, at different delayed time from the excited state, time, laser excitation process. So we can see if we have phosphorescence, delayed fluorescence, or the phosphorescence process. In the case of this compound, which I showed here, you can see that first you can see that our blue curve, blue spectra, is the phosphorescence, is the delayed emission at 80 kelvins, which means we froze our molecules to avoid that it will move, and we would see some delayed emission or reverse intersystem process. If we would heat up our molecule, we have our prompt fluorescence, so very fast nanosecond fluorescence, which is here. You see the red curve, red spectra. And if we go to the same delay time where we saw phosphorescence, but at 300 K, so around room temperature, we see that our emission is almost the same like the fluorescence, and like I showed in the beginning, explained the beginning, that means we have delayed fluorescence process. And in the case of the other molecules, we have similar behavior. We have phosphorescence and our delayed fluorescence, not the TADF. Remember, delayed fluorescence process. From this analysis, we can only estimate if we have delayed fluorescence process. We cannot differentiate, even by changing the temperature, if we have TADF or TTA, because both delayed process are affected by the temperature and will rise with the temperature. So if your emission is rising, intensity is rising, if the temperature doesn't mean you have TADF. With TTA process, it will behave similarly. So we know that we have our phosphorescence, delayed fluorescence, and the prompt fluorescence. Moreover, if you will check how our molecule will behave, the same molecule, 300 Kelvin, so room temperature. If we will check our spectra at different delay time, you see that from the less than nanoseconds up to 20 milliseconds, the spectra is almost the same. So again, we have very nice delayed fluorescence. But if we'll cool down our 80 Kelvins, we see that in the beginning we have phosphorescence. Yes? Is there any question? Okay, I don't understand, so I will go through. So we have our phosphorescence and we have our prompt emission, so our fluorescence, and with further delays, we are getting our phosphorescence emission. So thanks to this technique, we can differentiate different emissive properties. Later, I will show you how to distinguish between fluorescence between TTA and the TADF. Thanks to this analysis, we can estimate if we have CT emission or we don't have CT emission, if we have delayed fluorescence or we don't have fluorescence, delayed fluorescence. If we think about the TADF emission, we have two different types of the emitters, CT-based emitters and non-CT-based emitters. So in the case of the CT-based emitters, those are usually ambipolar emitters. They have ambipolar characters. That's why we investigate that in the beginning. And how to get that? We need to have orthogonal conformation of our molecules. So they should be, like I would say, perpendicular the donor to the acceptor planes, like you see here. How the non-CT based TADF emitter looks like? So like I told you, where we have our localized emission, and in the spectroscopy, if in the polar solvent you will get exactly the same emission, which means we have non-CT emitter. Usually those are quite planar or symmetrical molecules, which has something called multi-resonance effect. Professor Hatakayama presented different molecules which had such effect, and they don't have ambipolar character. There are usual acceptors or the donors, and they also have TADF process. This is one of the examples of our molecule which had TADF, but they were not ambipolar. They were pure donor. They had donor character. The emission doesn't matter if that was the polar or non-polar solvent. They were quite similar, but we observed delayed fluorescence emission. And they had similar properties, emissive properties as normal, as the ambipolar one, but they were only donor molecules. Another important factor which you should know, we always explain our ST gap, so the different energy difference between singlet and triplet level, and this is good for screening. Nevertheless, it's not the actual energy which you need to put into your molecule to push from the triplet to the singlet state. Actual energy, it's characterized in a little bit different way. So we are taking your delayed fluorescence emission. So we are characterizing how your delayed fluorescence is changing at different temperatures. So here you have Arrhenius plot. We have emission in this way that we have spectra, which we are integrating delayed fluorescence spectra, and we are checking how the intensity, this integration intensity, is decreasing with the dropping of the temperature. So you have low temperature here, high temperature here. And using this Arrhenius plot, we know what's the minimum energy to transfer your triplets to the singlet state. And actually, if you look in the literature, usually it's like half or even less of your ST gap, which means the actual energy to have TADF or delayed fluorescence is much lower than your ST gap. Nevertheless, this is more sophisticated analysis. So for typical screening of the emitters when organic synthetic students or professors are synthesizing hundreds of them, we need to screen them much faster. And finally, to differentiate between the TADF process or TTA process, the only way to directly prove that is the, I would say, laser fluence analysis, which is again similar process. We are collecting our different delayed fluorescence emission. We are integrating our spectra, and we have our intensity. So we have intensity, and we are checking how it behaves at with increase of our excitation energy. In this case, laser pulse fluence. So we are increasing our laser energy and checking how what was the increase of our emission. If the power or the increase of this emission was in the power of 1, it means for one triplet state, harvested triplet state, we are getting one singlet state, and that means we have TADF. If the value will be close to 2, it means for 2 triplet states, we are getting one singlet state, which means we have TTA process. In many cases, when you are analyzing your molecules, the value is between 1 and 2, like 1.5 or so on. That means we have mixed emission, because it happens as well. But without this analysis, we cannot directly prove that we have TADF process. So that's one of the way of doing that. Maybe I'll go a little bit further. So CT or non-CT? Generally, it depends on your emitter. In the case of CT-based emitters, which I will show you later, we can tailor our emission. We can change our spectra by simply changing the host or changing the little bit conformation. I will show that, which help us. Also, the CT emitters are ambipolar, which means are easier to implement in the OLEDs, because they can already then beginning inject holes and the electrons. Where in the non-CT emitters, usually in most of the cases, we need a proper host to use them in the OLEDs. Nevertheless, most of the CT, also non-CT emitters, you can use as hyperfluorescence emitters. So it's not. So generally, it depends where you want to use them. Both of them has a pros and cons. So we are going now to the host management. You have your TADF emitter, and you need to know if there is a way to use this material. All the CT-based emitters, so we can say most of the TADF emitters are are influenced or are affected by the host. Where you are using them in the OLED, it's not that you are using them as a pure. Usually they are in the matrix, in the organic matrix, small molecules or PVKs, CBP, DPEPO. There are different matrices which are used for the OLEDs, usually depending on the emission, if that's blue, green, or red, but that doesn't matter here. And this host is affecting your molecule because of your charge transfer state. If you have higher polar host, then your CT emission or charge transfer state is dropping to the lower energy. Here is example of one molecule where in one case we have host as CBP and in other case we have Xanax matrix. If you look simply on the phosphorescence here, blue, and phosphorescence here, violet, the emission is completely different. The wavelength is completely different. Here the maximum was around 550, here is 600. So by simple changing of your host, we are pushing to the lower energy. So we are tailoring our emissions. What else? We can see that here the delayed fluorescence component, if you will integrate this value, it's the contribution of delayed fluorescence is much smaller than in the case of the polymer matrix. So there is another factor in the literature, DF-PF ratio. It's showing what is the delayed fluorescence contribution. So it's simple integration of this transient curve over the delayed fluorescence or prompt fluorescence emission, and you are dividing those values. Higher delayed fluorescence contribution, so you have higher efficiency, and in the end you have higher, more efficient emitters. So you see that changing simply host, we are affecting our molecule, our charge transfer state, and our final efficiency of the device. So like I said, in the non-polar host, if we have a high charge transfer state, doesn't matter if that's the singlet excited state or triplet excited state. If we will put that into the high polarity host, then we are dropping our energy. Our localized excited states are not affected, but we are affecting our charge transfer state. And in some cases, you can find the literature that our charge transfer state is moving below localized charge transfer state. And that can cause different problems, but simply you need to know that always your host is affecting your molecule. So when you are analyzing your emitter for the OLEDs, you always need to do the photophysics or the analysis in the same host which you used for the OLEDs. Because in some cases, if you are especially analyzing in the solvent and you are saying you have TADF there, it doesn't mean you will have TADF in the OLEDs. And other way around, you may not have TADF in your solvent, in toluene or THF when you are doing the screening, but you will have in the OLEDs. That's why host matter. The other factor is conformance, which we found during our analysis with my colleague, Professor Takeda from Osaka University. He sent us for analysis several different molecules. And what we found out that some of them had mechanochromic properties. In the beginning, we were not sure what is the reason of this mechanochromic process, but the mechanochromic process is the process where we are adding energy, I would say, in the form of the grinding or fuming, and our material change conformation. So if you will see here structure, we have acceptor and the donors. And depending on the conformation, if it's axial or axial equatorial or equatorial, we have different, in this case, of the emission of the solid sample. So we can have the equatorial, we are moving or increasing our CT nature. We are decreasing our emission energy and increasing wavelength. And we are increasing our CT nature and TADF properties of our molecule. So this is, you need to remember that in all those cases, it's the same compound. but it has different molecular structure in the different configuration, different conformation, which affect our final emission and final efficiency. So in this case, we have phenothiazine, and this phenothiazine has different configuration to the acceptor plane. And what is important, we can also tailor that. So we can boost one of the to form different conformers in our emissive layer. And here is example. Here I analyze different crystals, pure crystals, because we need to know how our emission behave for different conformers. So we had 3 different conformation: axial, equatorial, and equatorial, and we analyze how the emission change over the time, how the prompt emission delayed fluorescence looks like. So we observe here what is our now known, that in the case of the equatorial we have pure TADF emission. Usually ST gap is much smaller, where in the axial we observe mostly room temperature phosphorescence, so very efficient phosphorescence process at room temperature. And in this sorry, that will be later, because in this publication I present how to use solution processing to tailor our emission or to form proper conformation. Maybe we'll skip that because I'm talking too long. So as a follow-up work, we decide to focus how to tailor molecules which will have more different conformers, and how those different conformers will affect our emissions. So in the meantime, we had molecules which had 8 different conformation, and depending on conformation had different TADF or RTP process. In those times, we wanted to push for more phosphorescence, room temperature phosphorescence properties, and because of that, we decided to focus on the molecules to form more mixed axial equatorial conformation rather than equatorial one. And with Yohei, we designed more silicon-based derivatives, which showed really nice properties. And in this case, most of the conformers were phosphorescence-based conformers. Through the analysis, the amount of conformers were more than 35, as I remember, but we divide them into those 5 groups. So how to manage those conformers? When you have your molecules, you can manage again by host. So you are not affecting only your chart transfer state, but you can also affect conformation which is formed by evaporation, because in this case we want to have efficient OLED devices, so we are evaporating our molecules. And here is example that in the more polar solvent, polar host, we are forming an emitter which is purely TADF, and we have pure equatorial conformation. Nevertheless, if we put into the TCTA host, which is the carbazole triphenonamine derivative, we are forming conformers axial, which has had only room temperature phosphorescence properties, and even more difficult to analyze, it has room temperature phosphorescence from 2 different states, triplet states. So going closer, if you look closer, what happened here? In polymer Xanax, which was solution process, actually in this case, we had dual emission, room temperature phosphorescence, and the TADF process, which which how to say that, fight with each other who will be more efficient, but we had dual emission, and in this steady state as well we observe both TADF process and the room temperature phosphorescence. If we move to the deep EPO, we had pure TADF process, very long lived. But in our TCTA, carbazole derivative host, we observe very nice phosphorescence emission and the phosphorescence from the T2 state. So if you look here, our risk process was completely closed, forbidden, and we observe emission from two different triplet states by simple host management, by simply checking how it's affecting our molecule. As I said previously, we can also manage our conformations in the solid state using different solvents for the deposition, for the spin coating. So I spin coat different devices using different solvents, different polarity solvents, and then compare to our crystal, pure crystal conformance emission, and check how they behave and if that's the equatorial or axial conformer. If you want to, it's really nicely presented in the paper, but generally it affect our conformations, and you see that actual other way around, that it was in the solution photophysics. Toluene form very nice equatorial layer and very nice efficient devices, and more polar solvent caused troubles, and we had more axial conformations and less efficient devices. But generally, we proved that by solution process, by changing the solvent, we can affect our final conformation. Another way to modify our emitters, and the way which I really like, is the electrochemical modification. So we are going back to beginning, where we are analyzing our molecules and doing the cyclic voltamperometry. Here's an example for those three molecules which undergoes electrochemical processes. As a standard, or for comparison, we have one molecule where our I would say electropolymerization parts are closed by the ometoxic group. So we know that this molecule will be stable, and those two will undergo electrochemical processes. And we also present them as electrochromic windows and as the OLEDs, how they will behave. In the case of OLEDs, because that's the case of this whole presentation, if you look how they behave in the Xanax matrix, we have very nice phosphorescence, blue line in all the cases, and we have very nice delayed fluorescence process at room temperature 300 K. Very nice transient curves, so they behave like the TADF emitter, but like delayed fluorescence emitters. But if you look to the ST gaps, they are quite huge, so they are on the margin of the TADF process, and the emission was here a mixed emission, both TADF and TTA process, so they were not efficient like we wanted. So what we did in the first place, we changed host to the CBP. So very nice matrix used for OLEDs, and we check how they behave. Like we expected, our ST gap dropped, so we know that it will be more efficient for OLEDs to use CBP. Nevertheless, the gap is still high, and we want to drop this a little bit further. We want to have lower ST gap and have higher delayed fluorescence impact and contribution. What to do? Again, use electrochemical polymerization. I don't know if you already know how it looks like, but we are modifying our emitter on the electrode. We are applying potential, we are going to higher potential, and we are observing additional processes. If the process will be reversible, that means we don't have additional reaction. We would have, if we will have the reversible peak symmetrical, then we don't have additional reaction. But here you will see that we don't have reduction process and we have new peak, which means we are forming polymer. We are electropolymerizing our molecule. With every scan, every nuke cycle, our area is rising, which means we have our, I would say, volume of our polymer increase. So in every case, we have more and more electroactive species on our electrode. If we would form here something which is degraded, so inactive from the electrical point of view, our current will drop and we would see more like a flat line rather than very nice reversible increase here. And with every step, every cycle, we are growing more and more of our molecule of our polymer on the electrode. Then if we would take our polymer, dissolve, clean, of course we need to clean, we've got some residue, but if we'll dissolve that, process that, then we have very nice polymers or oligomers, depending how we'll process that, on the in solution or in our form to use. Here is example in those molecules how the electropolymerization process is going, how they connect with each other. In the anodic electropolymerization process, we are forming polymer through our donor. So we are increasing conjugation of our molecule, and we are dropping our band gap absorption and the emission of energy. But you need to remember that it's connected mostly with donor part. So if you think about our molecule like here, we are affecting our donors. Acceptor should remain. So everything which is associated with acceptor, like in our case or in most of the cases, phosphorescence process, phosphorescence emission, should remain, and our charge transfer state, which is associated with donor, should drop because will have higher conjugation and you should have higher emission wavelength, so lower energy. Like in this case where we'll get our polymer. Here is presentation of the processes where we are forming our polymer, which is increasing, but I will skip that. So what happened for our polymers, where we had our donor, we had our small molecules XNAP and XDAP, but in the polymer form, when we pair our phosphorescence and smaller, when in the monomer, if we'll call like that, we had like 0.4. Here we drop that half. If we'll go to the CBP, the same. Our ST gap drop, which means we got more and more efficient emitter. So like here in the table, if we'll compare actually our small molecules, so the monomer and the polymer, we drop our ST gap 4 times, and that's quite significant by simple electrochemical modification. What is also here interesting, that we affect a little bit more of our triplet state. Our triplet state rise a little bit. It shouldn't, but it seems so that it rise because we form actually the XNAP's molecule had naphthalene here, and what we found out that naphthalene work here as the steric hindrance and push a little bit the acceptor from the plane. So in the end, we made devices, we compared devices from small molecule and the polymer. And what we found out that we rise our efficiency significantly if we will compare small molecule of the SNAP and the polymer. So it rises like at least 3 times our efficiency of the devices, our EQE. So to conclude, we can use our experience. Doesn't matter if that's the chemistry or physics, and if we join that, increase efficiency of our emitters, our materials by changing our host, which affect our emitter, or changing conformation. We can tailor our materials. So in the end, actually, if you are organic chemist, doesn't matter what you will synthesize. If you will think you can tailor your emitter, you can always implement that as OLED, but you need to think how to use it. Thank you very much. Thank you for your attention.
Thank you, Professor Data, for your nice, wonderful talk. And we really enjoyed your talk. And quick couple of questions we can have from the audience. If you have anything, you can rise and then interact with the Professor Data.
वो प्याज लहसुन नहीं खाता है, वो मेडिटेशन करता है। हाँ, मंदिर में बोल देना चाहिए। मंदिर में भी मिल जाएगा। मेरा रूम में बनाना अभी और मेरा दोस्त है।
Professor, this room temperature phosphorescence, it looks like, I mean, deliberately it is not been made like confirmation, like it depends on the molecule it made, right? But there are certain molecules where deliberately people make which gives a room temperature phosphorescence.
Yes,
by having
so
pure yeah.
Yes, so those molecules, a couple molecules which I present, they were like designed to have room temperature phosphorescence contribution. We have now much more, which we already published. What we presented that it's not simple to synthesize them. In most of the cases, we need to think how not what the molecules have, but how the parts of molecules are organized to each other. So the conformation which needs to be stable. Yeah, but we can design now many different room temperature emitters, room temperature phosphorescence emitters.
Professor, in Chilchon, what are all the facilities that you have equipped in your university? Your group.
So what we have, we have a lot of different spectroscopy, typical spectroscopy like fluorescence, but we have also the laser systems which allow us for the delayed fluorescence analysis, and in the end for the devices to make and analyze devices. Yeah, so we can do from the synthetic point of view up to devices. Whatever is needed.
That's great. That's great, Professor. So, any one of you from audience, if you have any questions?
Hello, Professor Data.
Hello.
Yeah, this is Dr. Rajesh Shamle.
Ah, yes, I showed you a molecule. Yes, yeah. The one in the end about the electrochemical one. Yeah, we haven't published that yet because we modify our manuscript and yeah.
Yeah, correct. Nice to see you on this platform and a very wonderful lecture. And my question is that, see, that work phenazine, okay, coupled with the donor phenothiazine, correct? Related
Yes.
on mechanochromic, that conformation, when the conformation changes, like see the donors are on axial and equatorial. So can we tune the emission of that? See, like if the donors are at axial position, both the donors, okay? And if both the donors are not equatorial position, correct? So is there any change in or tune in emission, like see from green to blue or red to green, something like that?
But because I wasn't sure, did you do you think about particular molecule which has phenothiazine? Because generally phenothiazine by itself form conformers because of the sulfur position. So it's pushing out of the plane. So if you have any molecule which contain phenothiazine, you would have in the beginning probability of different conformation, and you can find that by simple, like we showed, mechanochromic properties, grinding, and other process, and tailor that in the end how it will behave in the emitter in the solid state, both by host or by the solution process. But it's not so easy, and it's not like you have pure conformer. Usually it's a mixture. Nevertheless, there is always some majority effect. So one of the conformers have most efficiency or most emission in this layer.
Yeah, so there is a when there is a good charge transfer, okay, so the emission goes to.
And I have one curiosity question. So how about the scalability of polymerization, that electrochemical polymerization?
Yes, that's a good question. So if you, to be honest, scalability is really high, because there are even equipments which you can buy which were used in like 80s to increase the electrochemical synthesis for polyaniline or PDOTs. So in French there was a lot of work on, in France, a lot of work on that. So scalability is possible. You can get a lot of such material. The problem is purification. So you can get a lot of polymer, but you know, with bigger equipment, you have bigger currents going there. So you are also getting higher weight polymers. And in my case, we are trying to push for more oligomeric structure when our weight distribution is more controllable, possible to control. Yes, so it's possible to scale up, but it's not so easy.
Oh, I see. And so you collect the polymer only that deposited on the electrode, and the solution may not be.
No, it depends how you will prepare that. You can also so in many cases, when you are doing electrochemical polymerization, of course, you are depositing that on the electrode. But a lot of material, a lot of oligomers are also dissolved. So depending which solvent you will use for your process, you can control which oligomers or polymers are going to the solution, remain the solution, which are deposited on the electrode. On the other hand, you can also design process where you will deposit your molecules, your polymers or oligomers on the electrode, and then you can dissolve in another solvent. You can also use rotating electrodes to decrease amount of deposited material on the electrode. So there are several. So as I said, it's not so easy. There are several possibilities depending on the materials and what you want to get.
I see. Okay. Okay. Yeah. Thank you, Professor.
You're welcome.
If there are no questions, please join. So you please join with me to thank Professor Data.
Thank you.
Thank you, Professor Data. We are looking forward in presence. I mean, next conference in India in person. So
I hope so.
I hope so.
Thank you. Bye.
Thank you so much. I'll write to you later, Professor. Thank you so much for your time.
Bye.
And all the audience participant, please join tomorrow morning 9 o'clock, 8:45. You can log in. There is a talk. 9 o'clock session will start. 9 o'clock we have a talk, Professor Par. Then 10 o'clock Professor Parameshwar Iyer, and 11 o'clock from Shekhar from ISCT Mumbai, and 11:45 Professor Anandan from NIT Trichy, And then we have a talk from Dr. Jayakumar from National Tsinghua University, Taiwan. In the afternoon session, 3:30, we have Professor Deepthi Gupta from IIT Mumbai. And we have 4:15, we have a talk from Magesh from SRM University, AP. Thank you. So we will meet on tomorrow morning, 8:45. Thank you so much for joining with us.
उसी टाइम बंद होता है या
उसी टाइम
चल रहा था
चल रहा है
ज्यादा दिन का ज्यादा
ज़्यादा दिन